wrong with this machine? Oh, this is because I'm recording it. I get it. Well, let's go. Come on, let's get that there, shall we? I don't want this on. Holy moly. What's wrong with this machine? I don't want to hear myself the whole time. What in the world here? I mean, isn't that what it's going to do the whole time?
Okay, so click.
What do I do here?
Click mute over here. Oh wait, no, just kidding. She wants me to hear. Okay, do this. Turn it off. There you go. Now you're good.
Goodness gracious, this is really something. She disappeared again. So I don't get this. Are you there, Bella? Can you hear me, Bella?
Yeah, I can.
Okay, good. All right. They're saying hello, so there you go. Good. Let's make. What if I made this smaller? Make it see. Oh, there we go. That's what I want right there. Okay, good, 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 good. All right. Finally. So here's what we want to do, and I know you know this, most of these three words. These three words, remember, refer to the solute concentration, not the solvent concentration. So there'll be a few things said here that at first they're going to go, huh? So we'll explain as we go here. The greater the impermanent solute concentration in a solution, the greater its osmotic pressure. Cell membranes usually are permeable to water, so it equilibrates by osmosis. Remember, that's just water. Say what? What I did? What I did?
He's asking what page we were on.
It's slide 37.
Okay.
Yep. So it makes osmotic pressure equal outside and in the cell. So let's see here. Isotonic. I got how many solutions to use that word? Isotonic. How many solutions do I have that I'm comparing? Two. I have to, right? I cannot have one solution that's isotonic. Why? Correct. So if it's an isotonic solution, there are two solutions and they are the same concentration. Let's go to this one. Hypertonic. Well, in this particular case, this is different. This one says I have a higher concentration of solute. Therefore, a high concentration of water. Last, hypotonic means I have a small concentration of solute, but a high concentration of water. Yes, see how that works? Good. So let's take these next couple of pictures and let's see what we can do with them. Chuck. This is just disgrace. This is a red blood cell. As you look at it, now we have a lot of light in here today. Does it seem to me bright in here, or is it?
Yeah. I think you should turn the lights off.
Do you want me to turn it down so you can see this picture? How's that?
That's a lot better.
All right, so this is a biconcave disc. So it's like taking a Tums, which is a flat disc, and squeezing it in the middle to make two dimples on both sides. That's what a red blood cell is. It is shaped that way purposely and designed, I like that word better, designed so that as it hits a capillary that is half its diameter. it can still go down it bifold. And no matter which way that cell hits, it can fold and go down the length of a capillary. So that's what one looks like. To look like that normally, which of the three solutions would this be? Pardon? Isotonic. Where are the two solutions? There's one outside the cell, but inside the cell is the other solution. So there's two solutions inside the cell, one outside the cell. What are they as they compare? You already told me, tell me again.
Isotonic.
They're isotonic, which means the same amount of water is going in as there is going out. Yes? Yes, sir. Yes, ma'am. Why is it a biconcave disc? Is that why you're asking?
Yeah, like what makes it like bi?
It is.
So why
You
is it-
see a dimple here on one side, but we can't see the other side.
Oh.
The other side is also dimpled. That's why it's a biconcave disc. We good? Yeah, let's see if we can just change your mind a little bit and see what we get here. The flaw they did here that irritates me is it's in what kind of solution in doing this? What would you guess? It's in a hypo or hyper? Can't hear you. It is in a hyper. So where's the hypo? Inside. Where is the highest concentration of water at? Inside or outside? So more water will go from the inside and go to the, so the cell overall will shrink. That word was crenate when we learned it the first time. What they did though is it shrank, and the guy taking the picture says, okay, let's get a good picture of this. And they blew it up to the same size the other one was. Not showing that this actually has shrunk. What are these parts right here? If it has shrunk, what are those things sticking out? No, membrane would need some constructive something to help it keep sticking out there. Do we have something like that? Here, let's try it this way. See my face?
I do.
What would I look like if I lost 70 pounds? This part here, what would this look like? Look the same?
No.
How would it look? Sunken in, so that would mean something sticking.
Out.
Well, that would be bone, skeleton, you're referring. Do we have any of that inside that cell? Yeah. Not bones, but it would be called the cell's cytoskeleton. Remember that? So we have with that actin fibers, we have microtubules, and we have intermediate fibers that are there that make up the skeleton of the cell. So if it's going to shrink, well, obviously, just like my bones stick out when I've lost weight, so will these. Yay, everybody? So what was the solution here that was inside the cell? Say it again for me. Hypotonic inside, and this cell was put into a hypertonic. Very good, you're catching it, okay? You gotta stop me, stop. How about this cell? What kind of solution? It's still a red blood cell, doesn't look like one, but it's still a red blood cell. What kind of solution did we put it into? Hypotonic. What is inside the cell? Hyper. Where's the highest concentration of water at? Outside. So more water will go in than goes out. So the cell's going to, and ultimately it will burst if there's enough of a difference between the hypotonic and the hyper inside. When I taught at College of DuPage, one of the labs that I did with students there for this subject matter was we would pick up, or I would pick up some veterinary blood from dogs, cats, whatever. And I'd bring it there to school. I had enough that I could fill everybody's test tubes with it. And what they had were several solutions, some that they thought were hypo, some were iso, hyper. And then we had some that we had solutions lined up that were just, well, this is hyper, it's a little more hyper, it's a little more hyper, and others are when it's a little less hypo, a little more hypo, a little more hypo. Y'all hear what I'm saying here? And here's what we did. We took a tube, a small tube, maybe that long, and kind of that skinny. And we would add maybe that much blood, but then we would add the solution to it, one of those. And then when the cells, you try to hold that test tube over these words, like yay, you see what I'm doing here? Yay. And I can't read through there. What's in my way? Why can't I read through? It's a red blood with a little solution added to it. Why can't I read through it? What's in my way? What's in the blood that's in my way? No solution. I can look right through the solution. Yeah, the blood cells themselves are in the way. So what would happen to my vision and looking at this if all of a sudden they all went bing and they all burst? Did I read through it then?
No.
Yes, because there are no longer any what there? No red blood cells. None. So as a result, I could see through it. So I had guys time their solutions, and there were some sitting there, still can't see. Are you with me there? So they were very close, but they didn't burst. You hear what I'm saying? So then we established, at least closely, what was the percentage of the solution they had in their hand by did it burst? The one before it did, the one after it didn't either. Yeah.
So like the cells aren't actually visible because they're so small, right? So it's like.
They are, but there's a whole, here's my test tube.
But
A
you
third of it was.
Can you like actually differentiate and see like? each
No,
cell?
no,
That's
no,
what I'm
no,
saying. It's
no.
so small, so it's like an opaque kind of. Like
It's a
thing,
red.
and then-
It's a red, but you can't see through
Yeah.
it. And it kind of looks, you're like, why can't I see? It all looks like it's a red dyed water test tube. But then all of a sudden you're like, they all of a sudden it's like somebody went, and now you see, well, now all those red blood cells out there. Yeah, I mean, Jim. Helpful? Okay, good. I think you gasped that probably better than you did with me last year. All right, good. Filtration. We have water moving in and out of cells all over your body, but we also have certain places where we have filtration. Process of forcing molecules through membranes due to pressure. Pressure in part, if you want to think of this first example, a heart pumping blood through arteries toward capillaries. Blood pressure due to the action of the heart is a type of hydrostatic pressure which is used to force, as a force for filtration. Filtration is used by the body to produce tissue fluid from blood plasma. In the blood, the first thing they'll do when they get a blood sample from people, at least some kinds, they spin it down. They put it into a centrifuge, and probably 45% of the tube, when they get it run and spun down, is red and white blood cells, platelets, etc. All that went to the bottom. At the top, it's kind of a straw-colored clear. So you look at that part that's clear. That's called what? So it's the blood plasma, but then because it's forced into this tiny, tiny tube called a capillary, some of that plasma goes out of the capillary and into the tissue. You don't call it plasma anymore. We call it tissue fluid. And you've seen tissue fluid. Y'all remember running and falling and skinning your knee? And then you pick at it. And you do that, and then all of a sudden you look, dead gum, it's not blood coming out. It's this straw-colored fluid. It dries up and makes a new what? Scab. Y'all hear me? That's what that stuff is. Water and small solutes are sent through the walls of capillary to deliver O2 and nutrients to the cell, while large particles like plasma proteins remain in the capillary. Now, look at this picture here. Yeah, look at it. See that picture? Wasn't that good? Did you guys like that one? I did too. If you look here, blood's going this way. And so as a result, we find there's a lot of blood pressure over here and less as we get farther from the heart. So notice where it's hit. Oh, wait a second. Here's a capillary cell, a capillary cell. There's one that's got a nucleus, but that one doesn't have it. There's one that's got one, but that doesn't have it. How come that one and that one and that one, and that, they don't have any nuclei in them? How come? Do they have them? Or don't they have them? This is a capillary. Capillary, all it is a tube with nothing but cells patchwork around to make the capillary. So I'm asking you a question here. These two don't have nuclei. How come? Oh, we have to open your eyes. Let's see if we can do that. How many dimensions is this material? Isn't it 3?
Real life.
Yeah, in real life.
Yeah. 3.
What is it's 3, but what is it there? So
Oh,
why
it's
don't those two there have nuclei?
Well, they do because it's circular.
Yeah, can you explain better?
I don't know if this is right, but like, so for instance, say the one on top far left, if it's like circular, top left,
Oh, here?
yeah, well, a little bit to the right. The one with the nuclear there. If it's circular, then the one below it does have it because it's just.
Well, if this cell is the same
Yeah.
cell here,
Isn't it?
right? It might be, there might be another one
Oh,
between them, I
well,
don't
that
know.
was my guess.
But my point is, when the knife went through like this, here it hit the nucleus, there it hit the nucleus, there it did not. There it did not, there it did not. Does that make sense to you now? All right, think of it just that way. And where these cells go like this together as a patchwork, there are places that are gaps. See that there? So where those gaps are, that's where that plasma is going out the hole, along with nutrients and amino acids and a whole bunch of other things, oxygen as well. So that's going out there, but all the larger things don't fit through those holes. So they don't. So far, so good. This is filtration. Try it again. You gotta get the next one. Oh, look at that, huh? We know that there's an active transport that we get some things by, but when we use active transport, we're not going from high concentration to low, we're going from low to high. That requires ATP, an energy source we use in the cell. So these are active mechanisms that require venous and triphosphate. Active transport, basically moving substance from an area of low to an area of high concentration through carrier molecules in the membranes. Carrier proteins can be called pumps since they move substances against the gradient. With the gradient is going from high to low. Low to high is up the gradient, okay? You don't want to do that and go like in the case of fusion, it goes large concentrations low. Says here as much as 40, almost half of the percentage of the cells and of all the ATP in it is used to do this process. Get things in that need to be in and out, need to be out. Substances moved by active transport, sodium, potassium, calcium, hydrogen, from sugars, amino acids, etc. All those substances move. Sometimes by this process, active transport. Now they're not exactly showing, perfect picture here, but you see this is kind of moving out. And so look, it's a higher concentration at the top. Inside the cell is a lower concentration. So we're going against the gradient. We're moving up. And as we do this, here's the part that's kind of cool in my estimation. So as you start there, notice this guy gets in a spot and he's got parts on him that's negative and positive and some that are hydrophilic and hydrophobic. that as soon as he hits this little notch, what's happening is some other hydrophobic, hydrophilic, positive, negatives are going, oh, I want to be next to that guy, and I don't, go turn this one. And all through this protein, a transformation, and here it is, it gets through. Now, that would mean that only a certain molecule with a certain positive, negative, with a certain hydrophilic, hydrophobic, or if you want to call it polar, non-polar, whichever you're comfortable with, is going to get through there. But I had to use energy because I'm going against the gradient. You guys are catching on? Yes, no? Or am I just up here chattering away?
I think so.
All right. We have big things, big, comparatively speaking, to the cell that have to get in. We got big things that's made that's got to get out. How do we handle those? Well, that's by endocytosis and exocytosis. Endocytosis says molecules too large to be transported by other means conveyed inside by making a vesicle that forms the cell membrane. This requires energy too, but we won't get into that. Exocytosis, the movement of materials out of the cell vesicle that fuse with the cell membrane and open up to the outside and secrete. Like I could say, I'm secreting sweat on my hand. That's coming out of cells and can collectively out of my skin right here. Now, there's three kinds of endocytosis. I think you probably know them all. Pinocytosis, phagocytosis, and receptor-mediated endocytosis. I think you know them all, but let's kind of highlight just cellular drinking is pinocytosis, cellular eating is phagocytosis. And in the case of receptor-mediated endocytosis, that's not just a something I'm bringing in. I want a specific sucker in and nothing else. Yay? So let's see what those look like here. So here is cellular eating, phagocytosis. There's a particle, and here's kind of the difference, the way I think of it. This is phagocytosis. Look at me. Pinocytosis, what happens? Watch my hand. This is the outside of the membrane. It just kind of invaginates in and it fills just like a balloon does and then shuts off. Now I have a vesicle inside. In the case of phagocytosis, literally parts of the membrane just go and just leap out around it. Now you don't see much of a leap here. It almost looks like it was poured into it, but it actually does come out of it. You can see it kind of goes up in this model that they're trying to draw here. Goes out, surrounds, now I got a vesicle right there. And I might attach lysosomes to that to bust it down a little farther to be used, but that's phagocytosis right there. This is receptor-mediated endocytosis. I want you to check this out. See these three proteins that are transmembrane proteins? Notice they have a certain shape to get those. Is that red, by the way, those colors?
Mhm.
Yeah. Get those red things that are out there in solution. If those red things get just at the right angle with the right speed, they'll stick into those suckers like this. And then once they're there, these things migrate whichever way, front, back, until they find a spot that this doesn't have. Right here should be a whole host of other proteins like this called clathrin. And if that's there, they'll go to that clathrin spot and that will just kind of sink in, pinch off, and then a lysosome or some other vesicle will come over, attach, and dismantle this connection, and then pull in those red jobs, sending these three guys back to the membrane. Y'all see that there? Yay? All right, cool. Now, we're getting a excellent thing from your book here again. I've told you this before, and I think you believe me now. It puts together similarities and differences that you can read in a very short moment. The differences between certain things that you need to know. There's one, there's another. And now we're on to the last part for this chapter, the cell cycle. So this is mitosis. I don't think we're going to do meiosis just yet here, probably later. And the chapters that are sexual reproduction, go back over a bit. So let's look and see what it says. a series of changes that cells go through when they divide. And the idea of divide is to have more cells. Main phases are interphase, mitosis, and cytokinesis. Now, is interphase part of mitosis? No. Mitosis is simply the nucleus dividing. Cytokinesis is who? Cells and organelles divide. Good. Interphase is a part the cell is in when it's not actively dividing. During the time, it has three parts to it, G1, S, and G2. That makes sense. And so as a result here, we have those parts. On this cycle, there are also checkpoints. There are 3 of them, but I think your book is only going to show us 1. These are places where it checks to make sure that everything is where it needs to be to work. So it says most cells do not divide continually. Can you come up with cells you know in your body do not divide? I got one right now I'm thinking of that hasn't since you were born. Which one do you think that is? Brain cells. When you lose brain cells due to an injury, a concussion, fever, anything like that, you're not getting those cells back. They're gone, and they're gone forever. So that means a part of whatever function they were a part of probably needs to move somewhere else on the brain surface. What about muscles? I have you look at me, but I don't want the girls to get excited. But, okay, so the idea here is you didn't know this, but the cells that you had as a young man, a young woman, they don't change. The number of cells, because remember, muscle cells in the first place, you can't tell where one begins and the other ends. So the idea there is, wow, that doesn't change either. All you do is either add more actin and myosin, or you remove some. So that kind of gets the idea. So here's the great couch potato today, right here. And there was a time I was much buffer than I am now. And so as a result, then I lost some actin and myosin. And in my past, I had much more, but my cell count for my muscles did not change. So there are cells that are like in that situation as well. Let's try this in here. You remember the cycle as you look at this here? From here all the way around into there is just one phase. What's an interphase? Interphase, and you see in this, interphase and mitosis are not the same. They're at different times doing different things. This is mitosis. From here to here is just cytokinesis. And here we have one of the 3 checkpoints, another one there and another one in the middle of this right about there. But they're only showing you the one. If things have been going well and you're properly nutritioned and your DNA hasn't been hit by ultraviolet radiation and messed it up at certain points, etc., then you'll pass that checkpoint. But there's a number of things you got to make sure that you pass before you get past. See those parts to interphase that are G1, S, and G2. Let's talk about those and see what they meant or what they wanted us to know about them. Come on, did I get this? Yeah. Interphase. Before division starts, in which the cell grows and synthesizes or makes new molecules, membranes, DNA, and organelles. This is a time of great synthetic activity rather than a rest of time. Most people see it because, like, geez, there's so much going on in mitosis and then cytokinesis, and then they're not doing it. What are they resting? No, they're doing cellular respiration. They're breaking down chemical substances. They're using ATP. They're doing, all of this stuff is all still going on. It's just they're not putting in dividing the cell. During the G1 and G2, basically gap or growth phase, it says phases of interphase. It says cell grows and structures other than DNA are duplicated. Yes, passes right away. During the S part of the phase of interphase, it says the DNA of the cell is replicated. That's the only time that it is. And remember why it needs to. I got one cell gonna become two cells, and I want an identical message in each in the form of DNA. So that's when it's replicated during the S phase. Almost done, guys. Can you hang in there a bit? What do you think? There we go. So, wait a minute. Oh yeah, two types of division, meiosis and mitosis. Meiosis is gonna make gametes. Remember that word? Gametes are sex cells. Sex cells are sperm and egg. And yes, you can say that in here because I think they're all adults. It ensures that those gametes have half the amount of DNA. So when they come together for us, it's 23 chromosomes here, 23 chromosomes here, come together for fertilization, it's now 46. Looking at, yes, yes. That's a different process that later will go over again in reproduction. This one, for now, more common type of cell division is mitosis, reduced to increase the number, basically used to increase the number of cells. Mitosis is division of the nucleus, cytokinesis, division of the cytoplasm. They take place at the same time. One overlaps the other, but they're two separate processes. One is dividing the nucleus, the other is dividing the cytoplasm and all its constituents. Regulated carefully so that each of the two daughter cells receives an exact, underline that word, exact copy of the mother's genetic material. But do we get an exact number of organelles in each side? No. Could one possibly be a little larger than the other? Yes. So cytokinesis isn't an exacting thing. Mitosis, as far as the nuclear material, is an exacting thing. All right, let's go through. So let's look at these phases. Mitosis is described in a series of 4 stages, but processes actually continuous. Now they're saying 4 in this book. I need to remind you of something. There are actually 5, and you learned them in my biology class. There's one that's called pro-metaphase. It's between prophase and metaphase. They did that because they felt like prophase had a lot going on. Let's make another phase. Does the cell actually know when it's in what phase or another? No. It just starts and finishes. We looked at it and said, let's put some organizations in. What sort of things happen first? What sort of things happen next? Well, let's call these this and let's call those that, and everybody will learn it that way. That's what it is. So it's just us looking at it and trying to organize what is it going on inside that cell. These that we've written down for prophase are probably pretty good, and you're going to see something about them. When I read these and I read telophase, they're the same, just backwards of each other. Watch and see. DNA condenses, that means it kind of coils up, becomes visible as chromosomes. Now these chromosomes have chromatids, sister chromatids, who are held together at a centromere. So that sounds familiar. Centrioles, that's not one we keyed on, but centrioles migrate to either end. They make the spindle that attaches to the sister chromatids to pull it out. Microtubules, oh yeah, that's the part of the cytoskeleton to organize the spindle. Nuclear membrane and the nucleolus disassemble, which means I don't what? I don't see them anymore. Now, check it out. Let's read telophase, skip ahead here. Telophase says, begins when chromosomes have completed their migrations to opposite poles. Yes. Nuclear envelope and nucleolus assemble, reach. Now I'm going to see them again. And it says spindle fibers disassemble. Chromosomes unwind and take back their chromatin form, which we can't see, even with a microscope. So now let's go back. Now that's the opposite of prophase. These other two, metaphase, the big deal there is everybody goes to the middle in a plane, not a line. Anaphase is when sister chromatids do what? They separate. Just as soon as you see daylight between them, that there's anaphase. Telophase we just told you about, and you're on the money for it. Now here's a picture. I want you to kind of take just a second with me to look at. So here we are in the late interphase. Can you still see a circle going around there? Yes or no? Yeah. So it has a nuclear membrane. I don't see this boy in there. Anybody else? Already the nucleolus has disappeared. Oh, does that have a nuclear membrane going around it like that? No, this is prophase. And first of all, too, I saw a little bit of, chromatid or something over here. Do you see it more? Yes. So now what we're going to do is go from prophase. We're going to go down here to metaphase and see these guys line up. There they are. And then that line is going to split. This is what the artist rendered it as, but you can see it here. So now we're on there, and now everything happens in reverse that happened here. Now we see these going to either end, yes. The spindle is going to disappear, yes. I even have the formation of this cleavage furrow where it's pinching off between. So this is the process that we have that is before us as far as mitosis is concerned. Now, there's parts now that I don't think you had before. No, one more thing and then that. Cytokinesis starts someplace in the midst of anaphase. It says here, begins during anaphase of mitosis when the cell membrane constricts around the middle of the cell. Constriction leaves this cleavage furrow. Y'all remember that cleavage I talked about? And every boy in the room went, remember that? Okay. So cleavage furrow is there. That's when the two pinch off. There's a contractile ring. You may have, I know you're not old enough to remember the Pillsbury Doughboy commercials. There are still some around, but there was one where he said, we have a fat-free donut, not that, what do they do? Biscuit. What do we have? A fat-free biscuit. And he goes, he pulls up this belt and he goes, he puts less weight on you and whatever. And he literally, if he just went a little farther, he would have been pinched in the two. Okay. It isn't on the outside of the cell that it's being sucked in. It's on the inside of the cell membrane. It's a ring of actin. And it just keeps getting smaller. Pieces of it start falling up and rejoin. And literally it just goes like that until the cells pinch. And it's an actin ring that does that on the inside. Varying amounts, it says here daughter cells may have a varying amount of cytoplasm, but they have identical genetic information. Here's what we didn't talk about in biology. I'm assuming no one else did. I know I didn't. The brain cell look like a muscle cell? Or do you think they look the same? What'd you say, sir?
Different.
Yeah, they're different. They're different because they perform different functions. So somewhere down the line, after fertilization, sperm and egg met, and the next cells and the next cells, and they divided, and all those cells looked identical to each other. But at some point down the line, there was a cell that divided and said one was going to be muscle cell and the other one's going to be bone. But they now differentiate or specialize, and they start looking like what they're supposed to be. Y'all hear what I'm saying here? That's differentiation. It says here, we humans have about 290 types of differentiated cells. That's all, just 290. Okay? Cell differentiation allows cells to specialize by using different parts of the complete genome that is present. Each cell and various types of cells. Some genes are turned on while others are turned off. So it says they're stem cells. What are they? They retain the ability to divide without specialization. They're still all the same. Their presence in the cell allows for continuous growth and renewal. A step down, progenitor. They're the daughters of the stem cells that are partially specialized. There's some things they can change and grow into, but there are others they could never be, supposedly. Stem cells, progenitor cells, allow cells to retain their ability to produce cells that will become differentiated cells in each area of the body. Let me give you a for instance. So there's your sperm and your egg. Here's your fertilized egg. They divide. more division, more division, more division, more division. All the cells we made so far are these. They could still become any one of the 290 cells that are different in us. Okay? But this guy, he's doing, we're gonna divide more, divide more, divide more. Now we got progenitor cells. This one can basically become a sebaceous gland type cell or skin cell, but nothing else. This is another progenitor cell that came from these progenitors. Well, it looks like it can make an astrocyte or it can make a neuron, but nothing else. Stem cells can become anything. Progenitor cells make sometimes more, make some things, not just these 2 or 3, maybe a few more, but that's all they can make. They can't make any of the other cells. So this is how it goes with respect, and this is the big hullabaloo about stem cells. The idea here is, gee, I have a liver that's going bad, and I've already had a transplant. And then when you take on a new organ that's not your own in a transplant, the problem is you got to take anti-rejection drugs. Anti-rejection drugs are known for their bad side effects. In fact, if you take the normal 2 that you take for a transplant, you probably need about 8 to 10 to 12 more drugs to suppress the 2 you took to suppress your immune system so that your body doesn't attack this new organ. Y'all hearing me? And you got to live with those side effects that these other 6 or 7 or 8 or 9 or 10 don't get rid of. So your quality of life is diminished. You're not right back where you started, slick as a whistle. You have issues and things you've got to balance every day, the rest of your life, however long that is. Apoptosis is what we call cellular death. Yes, ma'am.
Would that be the same thing if you got an organ removed?
One more time.
Would that be the same thing if you got an organ
The same thing, if that organ was a transplant, you mean?
removed? Yeah,
Or
so like, if,
pull this down just a notch so I can see it here. Go
like,
ahead.
if I donate like a kidney,
Yep.
like, there's no new organ coming, but there's organ leaving, is the recovery somewhere?
When the weekend that Mr. McCallum, he was 14. On the weekend, he was in Michigan at a camp with his youth group, which I think was Wheaton Bible. When he was there, a virus attacked his heart and went fast at it. And within 24 hours, he had to have a heart, and I mean now, or he was going to die. So there was one. There was a kid, not far from where he was, that had a motorcycle accident or some sort of something happened, and was the same age as he. So it wasn't like getting a 64-year-old man's heart like mine. It was someone his own age at the time. And so when he got that, the thing about it, it's the same age. We had tissue matched it. They had done other things with it to see if it was going to work first, be matched. But now he's got the heart, and it's been there for 26, 27 years. He is an outlier. Most people are dead. Well dead, long dead than him. Are you with me here? He is an extreme exception. Therefore, the blessing of life he has is truly a blessing. So the heart they took out of that kid to give him, that kid doesn't have it. But now imagine, he's still taking those anti-rejection drugs. What if we had the ability? We don't right now, but what if in the future, doing this kind of research for stem cells, we could have a heart made from his DNA? Are you hearing me here? Have it made, put into his chest. Now it's his new heart, and it's his. no anti-rejection drugs, no drugs to suppress the symptomatology and side effects of those drugs. You hear me? Does that answer your question? Sure, we'll try again then.
I was more asking like the reverse. So let's say Nadia needs a kidney.
Yeah.
And I'm perfectly able, and I give her one of my kidneys.
Yes,
What happens to me?
nothing.
Like my recovery.
Your
process.
recovery is quick and easy, and because this is an organ you have two of. But you can live with only 15% of kidney function. That's all your kidney needs to function as, 15%, and you can lead a normal life. So now you've taken 50% of your ability to her, you're at 50%. Your life's good. So that's the sort of thing that happens. You get nothing as far as any bad side effects.
Okay.
She, however, is now going to take those anti-rejection drugs the rest of her life. And any other drug she's got to take to suppress the bad side effects of taking those drugs. Does that make sense? We have cells that program for death. When you guys were in your mother's womb, what was between your fingers? Webs. Where'd they go? They're not there now. Where'd they go? These cells had to what? They had to die. So they had to somehow know to die. And it says here, the form of death is normally part of development rather than from injury or disease. Removes overgrown tissues, damaged cells. The steps in it go this way. The cell kind of rounds up from whatever its shape originally was, bulges a bit, nuclear membrane breaks down, chromatin condenses, enzymes cut up the chromosomes. Cell shatters into many membrane-bound pieces. Scavenger cells engulf and destroy the fragments. That is what happens to you or those cells that were originally the webbing of your toes and your hands. All right, we're done. Now, when's this bell ring anyway? You guys know? What time is it now? No way. It's done at 20. Raise your hand if you got your book. You got your lab book. Raise your hand. Yeah. All right, good. You can take a picture of mine. I'll put it out on the desk in a minute. So here's what I want us to do. And I want have all of you used effectively a microscope?
Yes.
Yes.
Confident?
Yes.
What power do you put your scope on to find something the first time? What power? The lowest power, and then move up. Okay, good. You know how the light works? You got two switches down there that make it less or make it more. All right, let me show you a couple things that I think you got just. Oh, wait a minute. You said it's over 20. Yep.
Yes. So
Well,
30
here's what
minutes.
you need to do. I'm not gonna try this today, but you see these on the floor are for you. You're gonna do the lab in these chairs. Going back. And then whoever remains, just kind of hump it down around one the corner. You are gonna read this lab 4, but you're gonna start, write this down and mark it. You're gonna begin on page 32, where it says, look at my finger, see my thumb right there on the page? See my thumb?
Yeah.
You see that? Can you see that?
Yes.
Okay, right there on that page, on 32, you're gonna start right there. And all we're gonna do is measure the size of what you see inside the field of view. You're gonna see this circle. How much is it? How wide is it at low power? And from that, we're gonna be able to determine how wide it is at medium power, and then how wide it is at high power. And the reason we're gonna do that is that every time you look at a microscope slide and look at cells, I'm gonna tell you, and the picture in your book's gonna say, what's the size? And they're gonna have to tell me that in micrometers on every single slide. There'll probably be around 20, 25 of them before the end of the semester. So you need to get size. And all the cells are close or around, but they're enough different that it marks them. In other words, you got a blood cell, that's probably a pretty small cell. Others could be huge. So we're going to figure that out by putting a ruler on the stage of your microscope. And you're going to tell me how wide it is. We're going to do a little multiplication division, and you're going to work out what it is for each one. And here's why. You need to remember or have written down in that book what it is at low, what it is at medium, what it is at high. So if I see it takes 6 of these cells to fit across, and I'm at 600 micrometers across, say at high power or whatever the number is, if I take 6 to fill the view, go across each one, we will divide 6 into 600 if that's the number. Each one's 100. You see what I'm saying there? But you first have to figure that out. We'll do that on Friday or whatever. What are we doing tomorrow? Tomorrow we'll do that. So read this lab. We're going to do that at the beginning, and then we're going to grade it. Put a number in for the grade. Then you're going to go to 5. Now 5 you can start tonight. 5 you can write answers in and do certain stuff because you know most of that information. But what I'm going to do is basically hand you a couple of slides and say, what are these cells? What's going on? What do you see? All that sort of stuff. So read all 5. Yes.
So what part of 4 is tomorrow?
All of it. But we'll do that together. That part, that lab, we'll do together. And what you want out of it is you're going to answer all the questions at the end of that lab.
The lab report?
Yes, where it says lab report. You're going to answer those questions once you find out that first number.
And that's the lab report.
You can do it, but if no one finishes in time or not enough, I'll wait and do it another time. I'm not concerned so much as when it's done, so long as it is done before you start 5. That makes sense? Now, here's what I'd like us all to do. Hey, wait a minute. What time is that?
It's 5 minutes to 1.
And when are we done? You got enough time to do this.
I said 30 minutes.
Yeah, you have enough time. I thought, I was looking at the hour hand going, there's no time. So here's what you're gonna do. Get a microscope, get a cord, plug in, go. Back there. All the way in here, guys, here. Microscope are in there, and so is the cord. Bella, here's the interesting thing for you. Can you hear me? Can you got it? Can you see me?
Yeah.
Lab 4 and Lab 5, I don't know when you will return, but that will be the labs we're going to be doing tomorrow. What? You can, just one to a table. When they fill up, you're done. So Kay has got hers, you got hers, you got 3 left, and then everybody else goes in the seats. The number we're going to get tomorrow, we'll just tell you what it is if you're still at home. And then the answers you can work out yourself in Lab 4 and Lab 5. And whatever little bit you don't see when you return, I'll just have you do it. Now, is that possible? You just out today, or what are you thinking? How long are you out for? Bella? Hey, Bella, you froze. Okay, when you come back, let me know. Oh my. All right, I can have 6 back there, but then otherwise, go to your seat and you'll be able to find a place to plug in. place your hand, those of you sitting in these seats, make sure you got your hand on it, turn that sucker around. Because you remember how you look into it, you don't have the arm facing you, you have the stage facing you. Okay, so there you go, you're good. And then the rulers, do you see the rulers right there on that middle table there? Your job before the bell rings is to figure out how wide it is at low power. But you got some obstacles to fill too. They're not going to be right on as far as focus. You'll need to do that. And then you'll need to count. I mean, you might think it's simple. But it's not. Yeah, one. Low power, look at me with her here. Look at it. Come on, low power here, quick. There it is. Yeah. Then what you're going to do is you see this knob I got? I'll roll it towards this. And I'm just going to lay this right on that metal so it's going to stay on the metal side. Like that. Now what I'm going to do is I'm going to match the light, etc. I'm going to look in there and see if I can find a line. Each line. Millimeter. And they're going to tell me how wide it is down to probably the second decimal. You can real extrapolation there or guessing. Millimeter first, but then we're going to do it then. Remember, you're just going to lay that on there. You're going to lay it on there. Move your knob. Get it in over now. Now I'm going to do a little drawing here in a second. Give me a minute to get it drawn here. Just a second. When you look at those little tiny millimeter lines, they're really small on to the eye when you hold it, right? Well, look at this though. When you look at it in the microscope, they're fatter than fat. Here's a question, all of you. Look up here. How far is it from there to there? How wide is that? How long is that line? I just went millimeter. Wrong. Wrong. Wrong.
Not a full
It's
millimeter.
an inch? What if I instead
Well, you have to go
said
from the
this? Now, wait a second.
half of the person. Yeah, there you go. Now you got
What is that distance from there to there?
a millimeter.
That's a millimeter.
Yeah.
So you have to make sure you understand you're including this thickness, which is the millimeter. So if I started there or I started here, each of these would be about a millimeter. So what you need to do, watch, you got a circle and you're going to try to put that circle right on that edge there. Are you with me here? Or if you're going to put it in the middle, or you're going to put it on this side, whichever one you want to do. But then I want to know this distance exactly with the best guess you can give me in decimals. Got it? Do that now. Move the knob. Yeah, or the bottom one. And then get it in focus. You want that thing in a perfect focus so you can see each little dot.
Oh.
It's pink, that makes that solid.
So where would your
Look in the part of the end of the lab where I think it's just part A, part B. Is that a drink? That's not microscope.
No,
Are you sure?
no, there's nothing over there.
Oh, okay. Where's that going?
So I can't bring it back here?
That needs to go in the sink right there.
This?
Yeah.
It was sitting right there.
Well, it's not supposed to be in the room. So just set it right there.
Mr. Ellis.
Put it right here.
Wait, can I chug it really quick?
Put it right
I
here.
can't chug it?
There. Now you can drink that at the end of the period.
Oh, I thought you meant over this. Okay.
Figure out that little silver screw, hold that tight. Back there. So, there is. It is 1,000.
So for this, I don't like the lowest I think, so I should schedule it today.
So, we have at the 10-minute mark, we're going to put the stuff away and then be responsible for cleaning the floor. You know what? Is there any way I can get these clean when you're done? I had to go and clean them each, but what be nice when you're done, don't put your microscopes away till I tell you. I think that's my advice right now. Yes,
Can you remind me what the difference between a millimeter and a micrometer is?
what's this?
Can you remind me the difference between a millimeter and
1,000 mic. One.
1,000 micrometers? What is that?
One. No. This one. Yeah. at this.
No, what?
Don't shut that out.
Look at this.
You don't need to, I mean, like, just concentrate on skinning, not anything else. Do that for another 10 seconds. Remove this and you'll be able to keep track. And I'll tell you, right about now, take your hand away. Okay? Are you right-handed? Okay, that's what you need to do, yes. Say something. Okay. Okay, how do I turn this thing off?